Hi, everyone. Thanks for joining in on this webinar organized by the Microbiome Task Force team of the H3Africa Consortium. I'm very happy to introduce you, Prof. Amibat, this afternoon, morning, depends on where you are. You're probably very familiar with her now from the brief bio included on the webinar flyer. So I will not go into all of that, but I'll say this, that Prof. Bat is passionate about what she does, as well as sharing that knowledge. She's an excellent researcher, a great teacher and mentor, and I'm looking forward to what she'll be sharing today. And in addition to Prof. Bat's presentation, we'll have the pleasure of listening to a report on some of the work Dylan McGinney, one of Prof. Amibat's students, has been working on, has been doing. So Dylan is an outstanding PhD student in the BAT lab, and I believe this promises to be an informative session. We'll be having a Q&A session afterwards as well. So you're welcome to type in your questions in the chat box or raise your hands during that time. Just some housekeeping rules. Please ensure that your mics are always muted during the presentations, except for when you have to ask questions afterwards. Thanks again for joining in. Ami, please go ahead. Yeah.
All right, thank you very much, Foke. It's really nice to see you. Good afternoon and good morning to folks from different time zones. I'm going to go ahead and load up my slides. And as Foke mentioned, I'll be giving the introductory part, where I'll talk a little bit about an overview of the microbiome, and then I'll tell you a little bit about some projects that we have ongoing in the lab. Then Dylan will be speaking, and then I hope we'll have the opportunity to have a great discussion about issues such as study design, making choices about studies, and also thinking about methods that you can apply. So the title of my presentation is From Precise Microbial Genomics to Precision Medicine. This is the most important slide in my presentation, a picture of many of the fantastic people in my lab who have carried out much of the work that I'm going to be able to share with you today. If you're interested in learning more about our lab and what we're working on right now, please feel free to visit our website and also check out preprints that have unpublished data that are exciting and emerging from our group. So as you all know, we live in symbiosis with bacteria, viruses, fungi, protists, and even archaea. The way in which we live in symbiosis with these organisms is often at interfaces like this one. This is the interface of the colon. This happens to be the colon of a mouse, but if we were to take a look at a picture of the colon or large intestine of the human, it would look very similar. Up on the top left, you have the stool compartment or the fecal compartment, and you can see many different bacteria that are present. They've been fluorescently labeled in this particular case. On the bottom right-hand side, you can see the human cells that are DAPI counterstained in blue. And then between the human and the microbes are a couple of layers of green, and those are thick and thin layers of mucus. Now, what we know to date is that the microbiome can be very important for many physiologic functions of the host, ranging from barrier protection, how well this epithelial barrier works, to immune modulation, to infection, and even nutrient extraction. So the microbiome plays a lot of important roles for the host. And for the most part, the thousands and thousands of papers that have focused on the microbiome that have been published to date have focused on who's there in the microbiome. So they try to measure which bacterial species are present in the microbiome, and they try to link that to these physiological functions in the bottom right. Some effort has been placed on asking questions of what these microbes make that might be involved in crosstalk, but there are many fewer studies on what they make as opposed to who they are. And at the end of the day, our entire field seeks to try and understand this crosstalk that happens between microbes and the host. If I summarize a whole lot of literature together, what we know is that microbes and humans affect one another both positively and negatively. And when one changes, so does the other. But to understand microbes, as with any scientific discipline, at the cornerstone of what we do is asking how we're going to measure the thing that we're interested in studying. And in this particular case, we're interested in how we can measure microbes. Answering that question, who's there? Now, you all know that traditionally in microbiology, we can study microbes by culture. This works very well for certain bacteria. We streak them out on petri dishes, different types of plates. We then look at them under the microscope. Maybe we stain them and we can describe them, such as being gram-negative or gram-positive, and also describing them based on their shape and motility. But of course, this depends on being able to isolate and culture the organisms. And many of the organisms that we study in the gut microbiome in particular are actually pretty difficult to culture. Maybe not impossible, but difficult, meaning that this is a very biasing step. Alternatively, we can also measure microbes by their DNA or RNA. This happens to be a human cell pictured here, but the idea is you can take a cell, you can extract DNA, you can then sequence it, and you can understand what's there. But once you get sequence data from the microbiome, unlike when you're getting sequence data from human genomes, where you can just align to a reference database, in the case of getting a microbiome sample, let's say a fecal sample, you have to not only align these reads to one genomic database, but many. And when you do so, you can take these reads that you get from your DNA or RNA sequencing experiments, and you can align them to, let's say, the human genome, various viral, fungal, and other bacterial genomes. Now, in an ideal case, when you do this study, you're going to be able to describe every sequence that you get out of this shotgun metagenomics study or 16S study. Which brings me to the next point. When you apply sequencing in the microbiome, you also have your choice as to sequencing technique. You can apply 16S sequencing, as Voki did in some of her work that was published in BMC Microbiome last year. In this case, you can use 16S sequencing to amplify a conserved region within bacteria and archaea only. You can take that information and you can align it to 16S databases, and that kind of gives you a puzzle piece of the genome of any given organism. Now, strengths of this are that it's relatively cheap, high throughput, and you can amplify your signal, so it's good for low biomass samples. Disadvantages are, of course, that we can't get down to the strain level. Oftentimes, we can't even get down to species level classification with these data. The second problem is you miss organisms that don't have a 16S sequence, like viruses, fungi, protists, etc. So an alternative is to just sequence all of the DNA that's present, what I'll call full DNA sequencing or metagenomic sequencing. The problem then is that we don't have great reference databases, so you might want to just align to marker gene-based databases. This is what you would do if you were using a tool like MetaFlan. This allows you to achieve higher taxonomic classification, so sometimes you can get down to the species or even to the strain level, and you do get information about viruses, etc. Of course, you still only have part of the puzzle. So what our lab spent a lot of time thinking about is how can we take this metagenomic sequencing and get more information out of it? Well, the way we get more information out of it, honestly, is to put a lot more effort into it, both intellectually and, importantly, also computationally. When we do so, we can, for example, assemble sequences to try and get longer sequences as opposed to these short 100 or 150 base pair sequences. And we can get down to doing species and strain level classification. We get a good look at non-bacterial data, and we can even analyze pathways that are present. In so doing, we hope to apply these types of methods to better get to know our microbiome and to move beyond these kind of high-level numbers that I think many of us are familiar with. The idea that we have 10 to 100 trillion microbes in our body, they make up about 3% of our body mass. We have at least as many microbial cells as we do have human cells in our body, and there are over 3 million microbial genes. So the microbial genes far outnumber our human genes. So the summary of this very introductory piece before I get into some of our data is that microbes can be good, neutral, or damaging to the host. Most human-associated microbes are not dangerous. There are both culture-dependent and culture-independent, mostly sequencing ways to measure the microbiome. I didn't touch on it today, but you can also measure metabolites or proteins using metabolomic and metaproteomic methods. DNA sequencing has enabled a more comprehensive measurement of the microbiome, and 16S sequencing is very widely used, but sometimes has limitations. Finally, remember that there are more of them than there are of us in terms of cells and genes. So when we do these experiments where we sequence DNA from human gut microbiomes, the experiment doesn't look very much like the picture I showed you before, where every single sequence gets classified. Rather, it looks more like this, where some sequences are classified to human or other microbes, but a lot of data is actually unclassified. And that has a reason. It's because most microbes have yet to be discovered. And these are not just the microbes that are living in far-flung places, like at the top of volcanoes and at the bottom of seas. Even microbes within our guts and on our skins have yet to be discovered. And this is especially true for populations of individuals who have been understudied. Because of this, our lab and others have sought to find reference-free approaches to discover and track microbes. Put another way, if we take a look at this picture of fecal microbiota transplant capsules, these are used to treat people who have relapsed refractory Clostridium difficile colitis or a gut infection. We don't really know what's inside these capsules, or we sometimes like to call them crapsules. So how can we figure out what's in there without having to isolate and culture every single organism? Well, our lab has come up with two different methods to try and achieve genomes from metagenomes. Both of these methods are published, so I'm not going to go into great detail, and you're going to hear more about the nanopore-based method from Dylan after I speak. One question that we had early on was whether or not we could take these short reads that we get from Illumina-based sequencing and find ways to stitch them together so we can get more complete genomes. The idea is that we can go from these short reads, use de novo computational assembly approaches, and get full genomes. Now, as you might know, several groups attempted this with some pretty impressive success in late 2018 and early 2019. Their findings were published in these three papers, one in Cell and two in Nature. What they did was use these assembly-based approaches to generate what are called metagenome assembled genomes, or MAG, or MAGs for short. In these three papers alone, using predominantly published data, published metagenomic data, they were able to assemble 200,000 new metagenome assembled genomes. At that time, there were only 60,000 bacterial genomes in RefSeq, the reference database. And so as you can tell, with this approach, you can actually explore a lot of undiscovered territory. Now, these MAGs were a great step forward. They provided insight into uncultured organisms and represented an important step toward microbiome research that was reference-free. But of course, with any advance comes a catch, and the catch here was that MAGs were highly fragmented and incomplete. So here I am presenting a Circos plot, a circular representation of the genome of, in this case, a Phascolarctobacterium species, a MAG that was obtained from sequencing and assembly of a human fecal microbiome sample. Now, in this case, nothing was isolated and cultured. The human fecal sample was collected, DNA was isolated, the areas were assembled, and then we presented it here. As you can tell in the Circos plot, there are a lot of breaks between these green pieces, which are large contiguous pieces of DNA. And there are also these lighter green colored pieces that were not actually originally binned with the rest of these contigs. So these light green regions were left out of the assembly. In our first attempt to get better genomes from metagenomes, we used a platform that's developed by the 10x Genomics company called linked read sequencing. We call it read cloud sequencing, and we're able to generate much more contiguous genomes, as is shown on the outside track here in gold. This was work that was spearheaded by Eli Moss, a former graduate student in the lab, and Alex Bishara, a former graduate student who was joined with Serafim Betzoglu. Unfortunately, there was some bad news, which was while read clouds assembled some things really well, read clouds assembled some genomes very poorly. I'll ask you to pay attention to the gold dots here. You can see there are three different organisms, and they're arrayed based on their N50. N50 is a measure of genome contiguity, and the longer the better the contiguity, the higher the N50 is. When you have a perfectly assembled genome, the N50 equals the size of the genome. And remember, the size of the genomes are usually 2 to 6 or 7 megabases. So I'll call your attention to Prevotella copri here, which is one of the most prevalent and abundant microbes in humans on Earth. And you can see that our methods, despite their advances, did not substantially improve the N50 of this assembly. So that's when Eli Moss came together with Dylan McGeeney over time to come up with this alternative solution for complete genomes, which is nanopore sequencing and a lathe-based assembly approach. Dylan's going to speak to you a bit more about this, but if I just summarize, what they've been able to do with this approach is take these mixed microbial samples and get much more complete genomes, as is demonstrated on these outside rings of these Circos plots. So this is an approach that can really improve our ability to assemble genomes from metagenomes. So what can you do with this type of information? Well, one of the things that we've been interested in doing as it relates to precision medicine and health is track microbes. An issue that we have in the hospital here in the United States, that you also have wherever you are, is that a lot of patients in the hospital have bloodstream bacterial infections, meaning bacteria have gotten into their bloodstream and are causing infections. One of the questions that Tessa Anderman and Fiona Tamburini, former members of the lab, had was whether or not the gut microbiome, since it's teeming with trillions of microbes, might be a major reservoir of pathogens that escape the gut and make it into the bloodstream, especially in patients who are hospitalized for a long time and those that are undergoing HCT, or hematopoietic cell transplantation. This is also known as stem cell transplant or bone marrow transplant, and it's a treatment for people who have refractory diseases like leukemia. I won't go into the details here for time, but if you're interested in this work, it was published in Nature Medicine in 2018. And to make a long story short, what they did was collect stool samples from a lot of patients, as well as bloodstream bacterial isolate samples from when they got their infections. They then sequenced both of these sample types and compared the DNA sequences that they found in the two sample types. What they were able to find with these data was that 40% of the bloodstream infections that were found in these patients were traceable to a gut microbe at a time point prior to the infection, suggesting that the gut may be the place where these pathogens were hiding until they were able to make it out into the bloodstream. So if this is the case, if the gut microbiome is a major place where people actually harbor these microbes, another question that we might ask is, how do those pathogens get into somebody's gut microbiome to begin with? Put another way, when and where do patients catch their pathogenic microbes? To answer this question, Ben Siranoshan, a current graduate student, teamed up with Hua Tang and Niaz Benai. Hua is a professor of biostatistics, and Niaz runs our clinical micro department. This work is also available in preprint form online. And to summarize, we mapped sequencing reads from about 400 samples against 1,500 unique species-level genomes that we assembled ourselves. We then compared the strain populations between all pairs of patients. I'll just share with you two vignettes here. In one case, this patient 11342, who's on the top row, was able to transmit an E-thecium to patient 11349, who's on the bottom row. This happened during a period of time where they were overlapping, not only in the hospital at the same time, but actually they were roommates in the same room, suggesting that temporal and geographic proximity is a risk factor for transmission. This happens not only for microbes that are pathogenic, such as Enterococcus faecium, can also happen for organisms that are healthy or neutral for us, commensals such as Hungatella hathawayii, which you may know as Clostridium hathawayii. It was recently renamed. In the same sort of example as the one I showed you before, patient 11639 on the top had Hungatella hathawayii in their gut microbiome, as you can see in these orange squares. When they overlapped, as you can see by this little black square here, with patient 11662, that was actually enough to have a transmission event of this Hungatella hathawayii to this patient 11662. Now, it turned out that this patient on the bottom actually had a little bit of Hungatella hathawayii here at point 0. So the question was, is this Hungatella hathawayii at time point 58 the same as the one that the patient originally had, Or is it the same as its roommate? When we compared the two genomes of the organisms, I won't go into this in great detail, it turns out that we think that the patient number 2 actually had a different Hungatella Hathaway than the one they originally did. And it was very closely related to the one that the roommate had, suggesting a true transmission event. So at this point in our lab, we're thinking a lot about how we can modify the gut microbiome to decrease these infections. And we've carried out some early phase clinical trials. Once again, this work was just posted online as well, so I encourage you to check it out. In the last couple of minutes, I'll talk a bit about small proteins and why we're studying them. So I've talked a lot about who's there in the microbiome and how that might impact host health. I hope I've convinced you with some of these advanced sequencing and analysis methods, we can understand who's there at a much higher resolution. We certainly think that strains matter, and now we have a way to measure them. But of course, we're also interested in what they make. And the reason we're interested in what they make is because these molecules may be able to directly transmit across this barrier and signal to the host. While relatively more attention has been paid to small molecules that microbes make and how they can transmit across a barrier and communicate with the host, relatively less attention has been paid to proteins. In particular, Gila Sparrow, a talented former postdoctoral fellow in the lab, was interested in annotating small open reading frames that encode small proteins because these small proteins may be secreted and then diffuse across this membrane to signal. In this comparative genomics effort, this is work that was published about 2 years ago, we were able to identify over 4,500 new families of proteins, most of which were of unknown function, and about 30% of which we predict to be involved in cell-cell communication. To figure out if these proteins were actually transcribed and translated, Brian Freeman, a former graduate student in the lab, developed a molecular approach that measures transcripts that are being actively translated. It sequences transcripts that are actually bound by the ribosome in microbiome mixtures, and we call this method MetaRiboseq, obviously named after riboseq. In doing so, Brian was able to validate over 1,000 of these small open reading frame predictions, suggesting that these small open reading frames truly are transcribed and translated. Now that we have these pieces of information, it would be really nice to be able to annotate these small open reading frames in genomes and metagenomes. Part of the reason we hope to do this is because usually these small open reading frames are not annotated, which is why we went through this trouble to begin with. Matt Durant, a former graduate student in the lab, wrote a computational tool that's freely available, and it's also been published, that's called SMOREfinder, and it uses profile HMM and deep learning approaches to automatically annotate small open reading frames. There's a GUI version of this program, and I encourage you to take a look at it. At this point in the lab, we're interested in asking how microbes might use small proteins to communicate, and this is work that is actively underway and is a story very much in progress. So I hope I've convinced you that precisely measuring microbes is possible. The thing that we're working on is trying to figure out how these more precise measurements can help us enable more precise medicine. I talked to you about how we generate genomes from metagenomes, how we track microbes, and small proteins and why we're interested in them. But one of the things that we know overall is that the microbiome varies a lot from place to place. In fact, my human genome is much more similar to every other human genome on this earth than my microbiome is. Microbiomes vary dramatically, and things like lifestyle, diet, and environmental exposures impact microbiome assembly in early life and throughout life. The reality is, you and I both know that lifestyles and exposures vary globally in a very dramatic way, which means that to understand the microbiome of humans that live all over the world, we need to study humans that live all over the world. Unfortunately, there is very uneven attention paid to both human genomics, but importantly, microbiome genomics across the world. And if we take a look at distribution of publications focused on the microbiome of Africans, this is a little bit dated. It's from a couple of years ago from a review that we wrote together with Vokey and Scott Hazelhurst and others. You can see that this heat map ranges from 1 to 11 publications, and the majority of African, or many African countries, don't have a single microbiome study published about them. You can see that this number is increasing over time. We have on the x-axis years and on the y-axis number of publications, but we still have far too few published studies on this topic. Just to highlight this issue, I can ask you, well, what do we know about the microbiome of urban, high and middle income and low and middle income country dwellers? It turns out almost nothing. So in this picture on the left, this is from South America. This is a village of the rural Yanomami tribe. They live in rural Venezuela and Brazil. On the bottom right-hand side of this panel, you can see a picture of São Paulo, Brazil, which is one of the most populous cities in the world and the most populous city in Brazil. It turns out that there are many, many, many more studies of the rural Yanomami population than there are of people in São Paulo, Brazil. In fact, as of a couple of years ago, there were 0 publications on the gut microbiomes of individuals living in São Paulo, suggesting that we have a lot of gaps and holes in our understanding of the microbiome of people living around the world. So in 2015, I was invited by Michelle Berry and colleagues at the INDEX Consortium to come to the INDEX Consortium meeting. Michelle Berry, who's pictured here in the left, is our Dean for Global Health at Stanford University. And you can see here in this in-depth newsletter, Michelle Berry led a team of 6 senior faculty members from Stanford at the In-Depth Scientific Consortium. They included Mark Cullen, Steve Luby, myself, Eran Ben-David, and Marcela Alsan. This was when I had the chance to meet various members of the H3Africa Genomics Consortium. I actually heard them give a talk on the amazing work that the H3Africa Genomics Consortium was doing. And at the reception after the presentations, I approached them and asked about studies on the microbiome. It was with this that I was eventually introduced to Scott Hazelhurst, who is one of Vokey's mentors and now a great collaborator. And since 2015, we've collaborated very closely with the H3Africa Consortium to talk about how to advance microbiome work in the continent. So with that, I'll just thank you for your time, thank our lab, our collaborators, our many funding sources, and recent grads of the lab, and I'll pass it over to Dylan, who will speak for about the next 17 or 18 minutes, and then we'll have a conversation.
Great, thank you, Ami. I will go ahead and get set up here. Let's see. All right. And let me just make your faces small. Perfect. Okay, so I will be talking today a little bit more in depth about the project that Ami just introduced. As Ami said, I'm Dylan. I am a 3rd-year graduate student in the lab. I'm super excited to have the opportunity to speak to you all today and have a conversation afterwards. This has been a project I've been working on for the past couple of years, and I'm very excited to share it with you all. So as Ami discussed, the microbiome is this deeply dynamic and complex community of bacteria, fungi, viruses, archaea, protists, and more. And the microbiome is subject to a number of factors that alter what taxa can persist and thrive. And so as Ami said, this includes factors that are somewhat constant, like host genetics, which of course an individual's genome is going to be constant over time, but it also includes factors that change on a day-to-day or weekly or monthly basis. So diet, for example, is a very hallmark example of that, because it can strongly affect what organisms can persist in the gut based on what nutrients are available to them at a given time. And we know, of course, that there's huge variation in diet around the world. Additionally, there are factors like medications, especially antibiotics in particular, which can dramatically alter microbial composition. And then, of course, there are other environmental factors, for example, toxin and heavy metal exposures, Hellman's infection, that can drive changes in the gut. And so despite this complex network of factors that we know varies very dramatically around the world, again, as Ami noted, most studies that have focused on this human microbiome interface have really taken place in the global West, where populations are very industrialized, very urbanized, and where individuals consume just a very high degree of fatty or processed foods and have very easy and frequent access to antibiotics. And so this visual from Almeida et al. Summarizes the metagenome assembled genomes, which Ami introduced previously. So the metagenome assembled genomes that have been generated from microbiomes from different countries around the world. And as you can see here, the US, China, parts of Europe, all in red, have been really deeply studied with tens of thousands of microbial genomes generated from individuals in those different countries. And conversely, we see that there is this small minority of studies that have evaluated this other extreme of resource access. So you'll see Tanzania and Madagascar in particular have somewhere in the hundreds or thousands of metagenome assembled genomes. And those studies have largely focused on hunter-gatherer and agriculturalist communities in those locations. However, as is abundantly clear with this map, there's a huge disparity or gap in many countries around the world. And of course, again, as Ami presented, these really small numbers of studies at these extremes of lifestyle and resource access really can't be used to generalize to the majority of the world's populations that live between these extremes. And so we know from the small number of studies, such as those conducted with the Hadza, which is a community that relies primarily on hunting for food in Tanzania, we know that their gut microbiome varies when compared to communities with different diets and lifestyles. For example, Schnorr et al. In 2014 taxonomically profiled guts of the Hadza as compared to some individuals in Italy. And we see that those taxonomic compositions are quite distinct. For example, we see this red taxa here highlighted in the Hadza. I'll call that out for you here. And we see that that primarily corresponds to spirochetes, and that spirochetes are entirely absent in Italian guts. However, when we think back to that map of microbiome research, it becomes really clear that if we're identifying new taxa and different taxonomic composition in the Hadza, we're almost certainly missing a lot of microbiome novelty and different functions in communities around the globe. So I won't spend too much time on this since Ami stole my thunder here, but hearkening back to this literature review that members of the lab and Volke and Scott conducted, we identified only around 60 studies across 25 countries in Africa that looked at the gut microbiomes of individuals. However, if we narrow that down even farther and only look at studies that evaluate adults rather than infants or children, and studies that use shotgun sequencing rather than 16S sequencing or qPCR, we actually only found 5 studies across 5 countries in the entire continent. And so this underrepresentation of African adults means a few things. First, from a basic biology perspective, our field as a whole is really underappreciating the taxonomic and functional diversity of microbiomes across the continent. But more importantly, we are not evaluating whether the health and therapeutic findings that have been discussed in microbiome literature are even remotely applicable or relevant to these populations. And so this lack of research becomes a huge problem when we think about that direct relationship between the microbiome and human health. And so as communities undergo industrialization, they typically see, as charted here on the left, a decrease in infectious disease risk and an increase in non-communicable disease. So an increase in things like heart disease or obesity. And we know that the microbiome has a distinct relationship with the presence and severity of these diseases. Additionally, and perhaps more relevant right now, studies such as Harris et al. On the right-hand side have shown a difference in microbiome composition in individuals who respond in R or don't respond, NR, to various vaccines. So here, for example, they identified differentially abundant taxa between people who were non-responders or responders to rotavirus vaccines. And so altogether, this lack of comprehensive characterization of the gut microbiome across Africa really compromises our ability to assess the relevance and applicability of therapeutics for these populations. And so Ami began to introduce our research project, and now I'll delve more deeply into it. And so with our collaborators at WITS in Johannesburg, we are able to leverage some existing study sites from the AWIGEN project, focusing on two South African communities in our case. One is urban Soweto on the left and Bushbuck Ridge on the right. Soweto is an urban township that borders Johannesburg, while rural Bushbuck Ridge is a former apartheid homeland in northeastern South Africa. And you can take a look at the table here and very quickly see that Soweto is much more dense population-wise, and it also has an increased proportion of households with access to piped water and flush toilets. And so from these two communities, we focused on adult women, and we assembled a cohort of 190 adult women and collected single stool samples from each individual. These samples were then sequenced using both 16S and shotgun metagenomic sequencing. As Ami mentioned, Voke published a really beautiful paper on the 16S results last year, and today I'll focus on the shotgun sequencing results. And so after we classified our shotgun data, so after we took these sequencing reads and mapped them to reference databases to see which taxa were present, we found that the microbiomes from individuals in rural Bushbuck Ridge had significantly higher alpha diversity than their Soweto counterparts, which means that they had more taxa and greater diversity of different bacterial species than individuals in Soweto. And we somewhat expected this from prior works in the field that had observed decreased alpha diversity with increased industrialization. From there, we are able to identify a number of bacterial genera that were significantly enriched in one community over the other. So notably, I've highlighted in red here, Bushbuck Ridge is enriched for taxa like Succinatomonas and Succinovibrio, which are some of these hallmark genera that are lost with industrialization. And Soweto microbiomes in this lighter purple in the top were more enriched for taxa like Bifidobacterium and Bacteroides, which have been described as genera that tend to increase in abundance and prevalence with industrialization. And so moving forward, we sought to really contextualize these observations with those of communities at those extreme ends of lifestyle and resource access. So we used publicly available shotgun sequencing data for adult industrialized cohorts from the USA and from Sweden. And then we also used data from cohorts from rural Madagascar and from the Hadza hunter-gatherers in Tanzania. And so at a high level, when we compositionally compared these metagenomes, we found that the South African cohorts existed somewhat as an intermediate set of populations between the Swedish and American cohorts, which are pictured in blue, and the Tanzanian and Malagasy cohorts, which are pictured in red and pink. And so here we're just, every single dot represents a different individual from one of these cohorts. And we see that those South African individuals, interestingly enough, don't seem to cluster precisely linearly between those red and blue populations, but rather they seem to occupy this unique position in multidimensional space. And so what this might have started to make us think at the time is that there are probably some novel features, novel taxa, et cetera, that are present in these South African communities. And so from there, we sought to obtain a measure of how much unclassified diversity there might actually be in our South African cohorts. And so we quantified the proportion of our shotgun sequencing reads to different reference databases to see what % of those reads don't map to known taxa. And what we found here is that the less industrialized communities had a smaller proportion of reads that were able to be classified as compared to Sweden and the United States. So for example, we see that in the US, about 90 to 95% of reads are mappable to known taxa. Conversely, in Tanzania, that was more like 60 to 70%. And so this indicates that there exists taxa in these communities that have not been described, And at this point, we really wanted to get a glimpse into what those taxa might be. So we decided to perform de novo genome assembly on our metagenomic reads to try to build metagenome assembled genomes. And so to give us the best opportunity possible to generate really contiguous, not fragmented genomes, we decided to supplement our existing sequencing with a handful of additional nanopore sequencing experiments on some samples from Bushbuck Ridge. So I'll digress briefly here to explain a bit about nanopore sequencing. It's a technology that's new-ish. It started becoming very popular in the 2010s. And it is able to generate reads in the tens of thousands of kilobases as compared to typical shotgun sequencing, which generates reads that are usually 100 to 150 base pairs. And nanopore sequencing is performed on this small device pictured here on the left. It's a tiny instrument. It could fit in your pocket, which continually is mind-blowing to me. And what it does is uses a set of a few thousand tiny pores pictured here on the right to pull through individual native strands of DNA. And as that DNA moves through the pores, it causes variations in electrical signal that are then read out by the machine and converted back into DNA bases. And so the main advantages of this approach is that first, long reads make it much easier for us to build good genomes because it's much easier to put together a puzzle of a few big pieces rather than thousands of tiny pieces. And second, the portability of this nanopore system means that the sequencing could be conducted by us in our lab or in the future at whatever location or field site researchers are interested in. And so Ami hearkened to this earlier, but previous work in our lab led by Eli Moss developed both a DNA extraction and a computational processing workflow to generate these complete genomes from stool samples. And so from stool, we use an enzymatic extraction approach to break up and lyse bacteria and collect high molecular weight DNA. We nanopore sequence it and then use Eli's computational pipeline to convert that nanopore signal into DNA bases. We take those DNA reads and assemble them into longer fragments or contigs. And then from those contigs, we correct any errors in the sequencing and then check to see if any contigs are of a large enough scale to represent possible bacterial genomes. And so we were really excited to apply this to some Bushbuck Ridge samples and recover some really highly contiguous genomes for taxa that have few or even no public references available. So here I'm showing Circos plots. The contigs are represented as blocks of color along that circular axis with the nanopore contigs in green and the Illumina contigs in gray along that inner track. And so we were really excited to recover a single contig Treponema genome. Treponema are among those hallmark taxa that were originally thought to be lost with industrialization. They're incredibly difficult to isolate in culture, and that meant that our Treponema assembly was the first complete assembly from a human gut. We also assembled complete genomes for Bacteroides vulgaris and for a Prevotella species, and both of those are taxa that are globally really prevalent and abundant, but have very few complete genomes because their genome structures are very repetitive and difficult to assemble. We also observed a number of highly contiguous genome assemblies from organisms that have very low GC content. And so you can see for all of these genomes, they're in that 25 to 30% range, and that the Illumina assembly, so that shotgun assembly in gray on the inside, is very sparse and has very poor coverage of that full genome generated with Nanopore. And that was quite interesting to us. And so digging a little deeper, we observed that the GC content distribution of the sequencing reads themselves from the Nanopore and Illumina experiments were quite distinct. We see that the Nanopore read GC content in that darker gray has that distinct left skew. And so we can see that there's this higher density of reads at a low GC content for Nanopore, even from the same DNA extraction. So this really pointed out to us that Nanopore sequencing is able to unearth or uncover organisms that are on the lower end of that spectrum. And so importantly, these complete assemblies, beyond just checking off a box of having a complete genome for each organism, actually give us important insight into the genome structure and potential function of these new microbes. So here I'm showing a Lentisferia genome, and we have in this inner track here in purple highlighted different mobile genetic elements. So elements like bacteriophages, like transposases, that are able to move within and across bacterial genomes. And long read sequencing, by nature of those long reads, is able to very well place these repetitive elements into bacterial genomes. And that's a classically difficult feat for shotgun sequencing. And so here we see that a lot of these elements, like these integrated phages, are represented and encompassed in this green nanopore assembly, and we wouldn't have been able to detect them from the fragmented shotgun assembly. Extending this farther, we were able to identify and annotate all of these mobile genetic elements in nanopore genomes that were absent in their corresponding shotgun genomes. And we see across the board, this is the number of additional elements of each class present in nanopore genomes. And we're finding more transposases, bacteriophages, antibiotic resistance genes, all things that we might have missed from just the shotgun approach. So with that, I'll conclude by saying that we performed the largest gut microbiome shotgun sequencing study of adults in Africa to date. And we found that these microbiomes don't just exist on this simple linear continuum of industrialization. In fact, these microbiomes are exceedingly underrepresented in reference databases, which implies that there is a huge amount of novelty in these different communities that we've been ignoring. And nanopore sequencing provides one of just many different techniques that can help us better understand these microbiomes. And so our hope is that methods like this will provide a really strong foundation through which we can continue to explore the diversity of microbiomes of these understudied communities. With that, I will conclude and conclude and thank all of the collaborators. I'll go through this very fast. The Bot Lab as a whole, but with a special shout out to Fiona Tamburini, who really started this project and spearheaded it and was very gracious to let me hop on a couple of years ago. And of course, all of our collaborators in South Africa at each study site and at WITS, with a special shout out to Scott and to Voke, who is our host today, who have been just such excellent collaborators. With that, I think we're happy to take questions. Looking forward to chatting with all of you.
Excellent. Thanks, Ami and Dylan, for such excellent presentations. There is a question in the chat. I'll just start with that. And like we said at the beginning, please go ahead and type your questions in the chat. So just raise your hands if you do have a question. So starting with a question from Monica Mabazi. Is it right to say that some microbiota are pathogenic, or the pathogenesis comes in as a result of dysbiosis that leads to a shift in microbiota abundance?
So I'm happy to take that question. That's a great question, Monica, and actually one that has been debated very heavily in the microbiology field for decades. Stan Falco, who is a very famous microbiologist and a professor at Stanford, who unfortunately passed away a couple of years ago, has written very eloquently on the topic of, what makes a pathogen? Is it any bacterium or organism in the wrong place at the wrong time is pathogenic, or are there intrinsic things about a given organism that make it pathogenic? I would say that my perspective is that the truth is probably somewhere in between. There are organisms that have genes that make them capable of inherently being able to do destructive things to us. For example, there are organisms that have genes that can cause tissue invasion and destruction. And I would argue that those are probably pathogenic as opposed to being strictly commensal. These also can be kind of enhanced in terms of their abundance in the setting of dysbiosis. So oftentimes organisms that can survive harsh conditions can emerge as the dominant organisms when a gut microbiome is under pressure and becomes dysbiotic. And that's why I think sometimes pathogens not only are characterized by the ability to do bad things to our tissue, but also by their ability to withstand pressures and therefore become more likely to be in the wrong place at the wrong time.
Awesome. Thanks, Ami. Just to get to E. Kariuki, please go ahead and ask your question. Faisal, I see your question, but let's go ahead and hear from E. Kariuki first. Should you like to unmute?
Okay. Oh, I was unable to unmute in the beginning. Thank you. Okay, my question is also in regards to a new question I've seen in the on the charts about the nanopore reads. So I saw that I can call it a normal curve that was showing the ONT reads as well as the Illumina reads, and there was the propensity of the ONT being able to identify underrepresented some of the underrepresented sequences that are low in GC. And my question is, what is this attributed to? Is it because normally some of these approaches in Illumina require PCR-based approaches that enrich GC, or does it have any other underlying reason? Thank you.
Yeah, that is an excellent question and something we were very curious about. Our thought here is that a lot of the Illumina sequencing approaches, like you said, involve PCR steps, which can include bias, and a lot of them also include transposase steps, where there's a transposase that is used to insert tags onto sequences or into DNA. And there have been some papers that have shown that kits that use these transposase steps or actually have some GC bias, where they tend to skew towards higher GC regions, leading to much lower coverage of low GC. So that's our greatest hypothesis right now. I think it would be interesting to compare to a few different kits and see if that continues to be the case.
Thanks, Dylan. There's a question from... Okay, thanks, Dylan. There's a question from Faisal. I'm sure you can read it in the chat box, but I'll go ahead and just read it out for everyone. The tools for analyzing long reads are completely immature enough. How did you overcome this issue? Some recommend to include short reads and even synthetic reads to overcome the shortage of the current analysis tools.
Yeah, I can take that one as well. Certainly right that this is a very up and coming field, and so new tools and developments seem to be coming out every week. But I absolutely agree that one of the most major limitations of the long read sequencing platforms is that the reads that they generate are highly error prone. And so that means that, it depends on what study you follow, but that each read can have an error rate of 1 to 5% perhaps with the newest systems. And so that makes it very hard to have confidence in the genomes that you assemble, because they might have things that look like SNPs that really just come from read-based error. So the suggestion in the chat is exactly what we do in the lab. Generally, we assemble long reads and then we supplement with short read data, even with very low coverage short read data, to use that higher precision and accuracy of short reads to clean up our long read assemblies. Oxford Nanopore is starting to hint at the fact that that might be unnecessary as newer platforms come out that are less error prone. But for now, I think that that's the current field standard.
Awesome, thanks, Dylan. Edwin does have a question. Please go ahead and unmute. Edwin.
Yeah, thank you very much for your presentation, Dylan and Ami. My question is, during your studies, you compared some of your samples to some already existing cohorts from different continents. So my question is, already there might be some existing variation, between these continents in terms of their genomic, in terms of their genome and other variation. So the question is, how did you control for this variation, and how sure are we that what we are seeing is as a result of the differences in their microbiome, but not from the underlying variation that already existed between these cohorts?
Great. So I'm happy to take the first part of this question, then maybe Dylan will want to comment on our data in particular. A really nice paper by Daphna Rothschild et al. From Eran Segal's group in Israel examined this question of how much does human genome variation contribute to microbial genome variation. And the surprising answer was not a lot. This paper was published in Nature a couple of years ago. And what they did was they studied different ethnic groups that live in Israel. So as you know, Israel is a very small landmass, but because of its kind of political and social and demographic history, people from all over the world and many different genetic backgrounds came to Israel and live now a pretty homogeneous lifestyle. When they assessed both the genome and the microbiome of these individuals, they found that about 2 to 3% of the microbiome variation that they were able to measure seemed to be associated with the host genomic variation, suggesting that host genetics probably plays a relatively small role in kind of manipulating the composition of the microbiome compared to lifestyle, environmental, and other factors. So it's just an important thing to keep in mind, and it's part of why we feel so strongly that it's important to study the microbiomes of individuals living in various different lifestyles. Dylan, did you have anything to add?
Yeah, I guess I can note that confounding has certainly been a big concern for us and something that we've thought about quite a bit. And as far as experimental design and using publicly available data, there are certainly variations that are introduced often at the DNA extraction steps. And as we saw with the GC content results, even huge skews in abundance just from a sequencing method that you use. We've been really interested about that just within our own studies of comparing samples collected in different methods and using different degrees of preservation, and have found that at least with some of these stool preservation kits that they can be quite constant and replicable over time. So we're hoping to evaluate that a little bit more deeply. But yes, excellent point that it's something to keep in mind always.
Thanks, Amy and Dylan. And there's a question from Jambler. Hi there. How does the microbiome composition differ throughout the intestines, and how representative is our sampling?
Yeah, I'm happy to take this. So actually, it's a great question, right? We have microbes that live all the way along our intestinal tract or GI tract, from our mouth all the way down to the anus. There have been a few studies that have looked at biopsy samples of the mucosa of the GI tract at various different positions, including a study from 2018 that was published in Cell, again by the Segal and now Elinov groups. What they found was that the microbiome, the adherent microbiome, meaning the microbes that are stuck to the wall of the intestine, do vary over the length of the intestine. I anticipate that some exciting new work is going to come out on this soon, because there are now these capsules that people can swallow that collect microbial samples at different parts of the GI tract. And then they're excreted. Researchers then collect back the capsules and can take out the compartments separately and study them. So work by Casey Huang's lab at Stanford is investigating this. The graduate student who's leading that work is Rebecca Culver, and I anticipate that we'll see some cool results on that in the near future. I'll also just point out for all of you, I did put a note into the chat. If all of you can copy down that Google form link, or at least link out to it, this is a form that collects your ideas on or suggestions on study design and analytical tools, et cetera. I know that that was something that was of interest to many of you for this presentation. And since we won't have time to dig into it in great detail, I thought it would be great if you could fill out this form. Dylan and I will then summarize all of your answers, add some of our own thoughts, and then send around a summary report with Voki's help later on.
Awesome. Thanks, Amy and Dylan. We'll go through this very quickly. There are a few more questions or comments. Do you think a hybrid approach will improve things? I think this has kind of been answered already. And Miriam has a comment here. She says, hello, I agree that we should try to understand the functions performed by the microbiome and not only describe its composition, but on the other hand, I wonder if detecting the genes and their annotation is enough. The presence of a gene does not necessarily imply its expression. Do you have any comments on that?
Yeah, I think that's a great point. I mean, some things I'll suggest are that I think looking at transcription and translation is much more informative than looking at just the DNA sequence, because as you noted, the presence of a gene does not necessarily imply its expression. And so tools like MetaRiboseq can be helpful in that regard. One other thing to keep in mind is that only 15% of microbial genes are annotated and have a predicted function, which means that the vast majority of genes, we don't know what they do. So it's hard to assess what their contribution may be to function. And this is an area of active investigation around the world to try and improve that annotation.
Okay, great. Our time is fast spent, but we'll have just 2 questions. We're going to just go through very quickly, if that's okay, Ami and Dylan.
Yes.
Okay, so there's one from Paul Mbavu. How do we overcome contamination for the case of lung microbiomes, given that sample acquisitions in this part of the body picks up every microbe along the way?
Yeah, this is a great question and one that we have personally struggled with. We actually studied a bunch of bronchoalveolar lavage samples, which is basically when physicians put a tube down the throat, push saline into the lungs, and then collect it back up. And analyzing those data were very difficult. One, because they're low biomass. So kit contamination, laboratory contamination adds a lot to your signal, which is faint. And number 2, as was mentioned, you collect everything along the track. And so it's very hard to know what's at the bottom of the lungs versus what's in the upper airway. And the upper airway is very dirty, right? It's filled with all kinds of microbes from the oral cavity. So I will say that this is not a problem that we have solved. Careful controls can be helpful in that regard. So a case control study is advisable. If you're looking for really nice work on this and groups that are trying to advance this field, I would take a look at work by Frederick Bushman and Ron Coleman at the University of Pennsylvania. I think they've done some of the most thoughtful work in this regard, but I think this is one of the most difficult microbial niches to study.
Awesome, thanks, Ami. And one final question from Judith, which I think Dylan has actually answered. And I think that's an incredible paper, by the way, and it really speaks to this question. The question is, how long after migration do people's microbiome change? Weeks, months, years? Any additional comments, Dylan or Ami?
Yeah, Ami, I don't know if you have any addition to add besides sharing this paper, which was done by Dan Knight's group a couple years ago, and they studied immigration to the US by different Thai communities. But I'm scrolling through now and don't know if they provided a specific timeline for how quickly that's changing. But again, it's going to be affected by, by things that do not change with migration. For example, if you're maintaining the same diet pre and post migration, things like that will all affect that timeline.
Yeah, absolutely. This is a really good point, which Dylan brings up. Different communities have a different rate of change of their microbiome. If you move to a country and you fully assimilate into the diet and the lifestyle, and you live in a different way than you used to, then you're much more likely to have a microbiome that shifts very rapidly. Keeping in mind that we believe that there are probably some microbes that you inherit and transmit vertically that are not susceptible to changing quite so easily. So there's probably a core component of your microbiome that's inherited and is stable, and then a variable part of your microbiome that can shift with diet and lifestyle. There are additional studies that are actually coming out on this, some very nice work that you'll probably see coming out of Canada that has studied South Asian microbiomes as people migrate from South Asia to Canada. So I think you'll see even more work on this in the near future.
Awesome. Thanks again, Ami and Dylan, for such amazing talks and the questions from everyone and the answers we've gotten. And please do not forget to take some time out to fill out the questionnaire Ami has posted in the chat. It'll be very useful to each and every one of us, I'm sure. And just again, say thanks everyone for attending. And just as a plug for the Microbiome Task Force, we meet every 3rd Monday at 4 p.m. So you're welcome to join us. And if you want to be a part of our mailing list as well, you can send an email to Francis. Francis, you can go ahead and put your email quickly in the chat box, or just contact the office, the communication working group of H3Africa, and I'm sure they'll be able to push you across to us one way or the other. Thanks again, Ami. This has been amazing. Thanks, Dylan. This is really good. And yeah.
Thank you all. And it was a pleasure to meet you all. And we look forward to being added to the listserv also. Take care, and thank you, Voki, for organizing.
Sure, thanks. Bye, everyone.
Bye.